let's discuss the biological database. It is the collection of data that can be stored, searched, and retrieved. Data are arranged by the set of rules which are programs into the software that manage the data called as Database Management System or DBMS. Database help to easily handle and share the large amount of the data and suppose the large scale analysis by the easy access and the data updation. These databases are either proprietary rights or the public domain databases with free access. The information stored in these databases is either in the form of the flat files or it can be a relational database. What are the features of the biological database? Firstly, data is the heterogeneity, sequence, graphs, high dimensional data, shapes, temporary data, patterns, model data, scalar and the vector fields, extracted features data, high volume data, uncertainty, data curation, large scale data integration, data sharing, dynamic and subject to the computational changes. Let's discuss the classification of the data. We classify the biological data on the different bases. Firstly, we classify the biological data on the basis of the data source, which are divided into three: primary database, secondary database, and composed database. Primary database is the nucleic acid protein sequence and the structure information, which have the example gene bank, Ncbi, DDBJ, Swissport, PIR, PDB, or KEGG. Secondary databases which are derived from the primary databases, which have the examples Swissport, TREMBL, PFM, Prostate, Scope, Profiles, CAT. Composed database that collect and present the data after comparing and filtering them from the different primary database and exhibiting only the non-recurrent sequence. Example, AULO. Now, database classification on the basis of the type. First, let's genome database, sequence database, structure database, microarray database, chemical database, pathway database, enzyme database, disease database, and the literature database. On the basis of the data access, publicly available, available with copyright, browsing only, academic but not freely available, proprietary, commercial, restricted SQL queries against underlying database management system. Nucleotide sequence database की बात करें तो which are composed a group of the nucleotide entries, data repository that accept the nucleic acid sequence data and make it freely available to the public. Example की बात करें तो gene bank, EMBL, nucleotide sequence database and the DDBJ which have the data bank of the Japan are the major sequence repository from which various databases have been also derived. All these are the members of the International Nucleotide Sequence Database, concentrate and interchange the data. What is the International Sequence Database collaboration? These three databases has collaborated in since 1982 and each database collects and processes the new sequence data and relevant the biological information from the scientists in their region. Example, EMBLs collect from the Europe, GeneBank from the USA. These databases automatically updated each other with the new sequence collected from each region every 24 hours. The result is that they contain exactly the same information except for any sequence that had been added in last 24 hours. This is an important consideration in your choice of the database. If you can accurate and up-to-date information, you must search an up-to-date databases. Here is the Ncbi website, that is www.ncbi.nlm.nih.gov. EMBL की वेबसाइट है www.embl.de and full form of EMBL is the European Molecular Biological Laboratory UK. Database of Japan, that is the DNA database of the Japan. Site is ddbj.nig.ac.jp. Firstly, we discuss the gene bank of the Ncbi. Ncbi was established in November 4, 1988 as a part of the National Library of Medicine at the National Institute of the Health, USA. In October 1992, Ncbi, the largest biomedical research faculty in the world, took liability of the gene bank. Gene bank offer all publicly available nucleotide sequence, their protein translation, and their annotation information. Researchers from anywhere can submit their sequence and the data to the gene bank. An essential number is given to the submitted sequence and then released to the public database after the quality check. This is the gene bank site. Gene bank submission किस तरह से होता है? Bank IT is a worldwide web-based sequence submission tools which can submit the sequence directly to the gene bank. Another tool used is the scanning developed by the Ncbi. submission software available for the Windows, Unix and the Mac operating system. Sequence can use for the DDBJ and EMBL also. EMBL क्या है? European Bioinformatics Institute situated in United Kingdom provide the free access to the large collection of the nucleotide sequence along with the additional information stored. EMBL nucleotide sequence database first established in 1974. Various methods have been used to access the data, of which an important ones are the sequence retrieval system, that is the SRS, and FTP file transfer protocol. Webin is an interactive or many nucleotide sequence to the EMBL database. This is this website of the EMBL. Next, DDBJ, that is the DNA Data Bank of the Japan, was established in 1986 at the National Institute of the Genetics, Japan, with the support of the Ministry of the Education, Science, Sports, Culture, Japan. Several tools has been made developed at the DDBJ for the data retrieval and the analysis. And Sakura is a web-based sequence which is used to submission tool at DDBJ. And Sakura is used to interactively submit the sequence annotation as well as the information about submitters. This is the site of the DDBJ. Thank you.